my slide?
Yes, sir. Yes, sir. You can share, sir.
Sir, your slides are not visible, sir.
Yeah. Is it okay now?
No, sir. Still not visible, sir.
Yeah.
No, sir.
Not yet, sir.
Not yet, sir.
Yeah. Just a moment. Just a moment.
Yes, sir.
Now visible, sir.
Now visible, sir.
Visible?
Yes, sir.
Okay. So yeah, this is
Make a
this is
slideshow,
the,
sir.
is it visible now?
Slideshow mode haki, sir.
Full screen ಮಾಡಿ, sir. Slide
Okay.
show. Okay, okay.
Okay, sir.
Okay.
Okay, sir.
Yeah. Actually, it's a the bioresource. The refresher course is mainly on the bioresource, particularly using the genetic, the availability of natural genetic resource in India. So there are a lot of research is going on by using the bioresource which are been not much explored, and more focus is nowadays given by the researchers to explore these materials in order to implement in the form of some applications that can be useful for the societal impact. With this for this refresher course, I have planned my topic to deliver. It is related to utilization of plant resource for synthesis of nanomaterials and its application to sustainable crop improvement. As sir has introduced, I belong to Central University of Kerala from the Department of Environmental Science. Yeah. So when we talk about the recent trend of using crop against to overcome some biotic and abiotic stress, we are talking about a sustainable method of practice in order to reduce the impact of external factors that is causing major loss to the crop, particularly in terms of yield. So the recent trend is using various chemicals and fertilizers. You will have a lot of impact on its residues that remains in the soil for a longer time, and also at the same time the impact of these toxic residues will play a major role, not only in affecting the soil health, the microbiome that is present in the particular environment, as well as the beneficial effects that can incur on the plants, so that which will have an effective trend of getting chemical-free food materials for the population. As we know, that current trend of practicing the recent applications of what we call as the nanotechnology is a growing interdisciplinary area of studies which involves both material science and as well as bio nanoscience. These are the major two interdisciplinary aspects where the materials, either in the form of internal or external, is being coated in the three-dimensional phases of the biological material, so that its immune system of the materials can be enhanced to efficient manner, so that there will be no side effect on the system what we are adopting or what we are employing for. With this, there are various, medicine plants. India, as you know, it's a vast diversity country with lot of diversity in terms of our traditional, our food, our practice, including the nature. That includes our plants. And so far, as per the literature, we say about 13 to 14% of the plant materials have been explored, and remaining 80% plus has not been explored. out of 13% of the plant materials which has been known and documented, there are about 40 to 50% of the plants that is being documented in the form of medicinal properties. And these medicinal properties is a natural resource which are easily available with abundant, biomass that is been producing in certain regions where the local, particularly the localites, are using these as a traditional medicine for their practice in order to overcome various issues. So with this background, since we come from southern parts of India, we have a 3 major belt of Western Ghats, which is covered by the luxurious area where medicinal plants are being abundant in this area, and we want to have we need to utilize these plant materials for application. Why can't we try with the nanotechnology aspect? So as such, plant synthesized nanoparticles, as you know, it varies in the form and scale. And most important thing, what we synthesize the material from the natural source should be a durable one, so that the nanoparticles synthesized from other source can be easily comparable, and which can be a stable temporal activity can be checked in the plant system. And also it should have a durable resistance where it can easily taken into consider as a sustainable development. So in this way, our research is focused on nanotechnology by using the medicinal plants for the synthesis of nanoparticles. I use the word in the title nanomaterials because nanoparticles are those which can be used externally for the coating of the 3-dimensional shape where the object can be formed. In case of nanomaterials, we use various composites that can be used as a precursors or can be used as a capping agent that can stabilize the nanoparticles, and which can be used for the various practices, like we can either use in the form of priming or we can use either in the form of a spray. Most important thing, one I want to emphasize here is, in India, since we are in the developing country, as such, nanoparticles are not been allowed to use in the field level. So far, various documents are available. Yes, nanoparticles are very excellently, it is working, remarkable applications. Of course, we agree that are limited only to the laboratory. And if someone wants to have a nanoparticles or a nanofertilizers at the large field scale level, then we need some statutory bodies for approval so that it can be taken to the implementation at the field level. So this some policies which have been already made by the ICR system, and they have been working on particularly on nano pesticides and nano fertilizers. And as such, the documents is very clear that plant-based nanomaterials are the excellent commodities which can be used in the plant system to develop a sustainable crop improvement. Where in case of it can act against the biotic stress, it can act against abiotic stress, as well as the increased yield can be observed when you are having a nanomaterials in the form of priming or spraying on the plants. So as such, why the nanomaterials in the medicinal plant of the part of the plant, we know its importance, which acts as an healing property. In case of nanomaterials, if you synthesize the basic catalytic, electrolytic and mechanical, optical, antimicrobial, wound healing and anti-inflammatory properties of these nanomaterials are being considered as most significant. So with these positive notes, the more trend is now people are working on various nanoparticles, and that has been very much useful in the application part. And in recent years, as I was emphasizing on, metalloids or metallic metals. So most of the nanomaterials nowadays, there is a talk that nanoparticles are good. However, what about nanotoxins? So you know that the product which you are using as a precaution materials will be at the lesser concentration. I mean to say, it's a minimum concentration we use for the synthesis, but these applications, it is used in the wider form or in the larger scale. Of course, the nanotoxins issue may come in the future. And nowadays it has been already started. The debate has started that, okay, what about the nanotoxins? How you are overcome or reduce the nanotoxins? So keeping this mind, the metals which are very less and which are eco-friendly, which can not affect the nature. So those syntheses are the metal ions we are mostly considering in our studies. One is the copper, the next is the silver, then next we have the selenium. Of course, it's a little bit toxic, and this zinc. These are the major nanometals we are considering for synthesis of nanoparticles using various plants which have medicinal properties. And as I mentioned you already, it's a gaining momentum, a research which can produce a multifaceted properties, which includes antimicrobial properties, and also which can induce into the host system, which produces the durable resistance. Yeah, when I was using the plant utilization of plant materials that is available in India, we have taken the most available, which is very easy to collect, and these plants are very nearby, and it can be grown in. It is very easy to grow also in the garden, as well as it is easily available in the nature also. So some of the plant materials which we choose for the synthesis is we have already tested in our lab. The copper nanoparticles, zinc nanoparticles, selenium nanoparticles, and silver nanoparticles. And one such example is we have given the Bryophyllum pinnatum, which is very common plant, and it can be used for a excellent synthesis of nanoparticles, where we use for the individual nanoparticles as well as bimetallic nanoparticles also can be synthesized by using this plant, which has given a very excellent results in terms of its antimicrobial properties, in terms of its binding capacity as a priming agent, as well as it acts as an inside the immune system, how best it can trigger the cell wall or trigger the biochemical enzymes that can bring the resistance against the foreign entities that is attacking the host plant. Yeah. So this is the study where we have done for the bimetallic nanoparticles by using the host plant, model plant as a tomato, as the major bacterial wilt pathogen, that is Ralstonia solanacearum. And you can see, as I was mentioning you, when I talk about spatiotemporal durable resistance, previously it was known that only the chemicals have these properties. I mean the systemic chemicals. So these we just want to know whether these nanoparticles have the systemic nature in its applications, whether it can be primed in the seed, it can go inside the embryo, whether it can stimulate the emergence as well as the root as well as the shoot biomass. And we have also found that by priming the synthesized nanoparticles, that is plant-based, will also boost the emergence of the seedlings, as well as we got 3 to 4 days early emergence of the seedlings in the treated plants compared to the control plants. And we have also tested for this spatiotemporal induction of systemic resistance in the plants. We have done with the priming with the seed, as well as fortnightly we have sprayed the nanoparticles on the plant. This experiment mainly conducted in the greenhouse condition, and you can see the plant incorporated with the pathogen plus primed with the nanoparticles shows an excellent biomass, as well as has given a maximum protection against the Ralstonia solanacearum. And this protection was very much stable, and we have tested for its various mechanisms, starting from the cellular, then the enzymatic activity, then at the molecular transcriptome activity. We found that there is a good amount of genes that are upregulated, which are mainly corresponding to the resistance as well as reduction in the pathogenicity, that is virulence of the pathogen. So this is very clear-cut indications that similar, unlike our systemic fungicides, though have a promising applications or promising results, we have some residual effect and toxicity. And in order to overcome this, of course, I surely say that nanoparticles can be replaced, not at the very rapid way of applications, at the smaller rate, then go for the larger scale applications. Yeah, similarly, we have also tested by using the Fungamia seed extract for silver nanoparticles, and this is also quite significant results which we have achieved in terms of we conducted on cucumber, that is powdery mildew powdery mildew disease caused by the cucumber, which is a major pathogen. And since it is a semi-biotrophic pathogen, we could also see the aggressiveness of the zoospore. That means the arrest of the sporangia, whereby the release of the zoospores are completely reduced at more than 50 to 60 times as such compared to the control. This is also one of the significant results which we have achieved in our lab, that is using Pongamia seed extract by synthesizing with the AgNPS, that is silver nanoparticles. So I just was emphasizing on some of the metals which we have tested in the lab. Now, since the demand is more on nano fertilizers with multiple or multifaceted properties, now the Indian Council of Agriculture Research is promoting the researchers to work on the fertilizer point of aspects where they consider nano fertilizer. That can be application part also is very quite easy, and policy matters also not much hurdles in order to bring this product to the mainstream line. So in this way, the ICAR has very last 2022 they made a committee, and this committee had recommended the most of the biocontrol scientists to work on nano fertilizers instead of using other approach for reducing the pathogenicity or virulence in the host system. So in this way, they usually term most important nanoparticles that can be used is zinc oxide nanoparticles. So which they consider as a eco-friendly actions, because each plant requires the zinc for its metabolic activity and as well as its vegetative growth during all the stages. And if zinc nanoparticles are being supplemented in the form of nanoparticles, which remains in the soil and also improves the health of the soil, thereby there will be an indirect effect on the yield for the promotion. So this is what the concept says: eco-friendly action of zinc oxide nanoparticles. Not only the promotion of the biomass or the plant growth and other parameters, it also significantly suppresses the disease-causing microbes. This is most important. Microbes in the sense, it's a broad spectrum where antimicrobial, antifungal activities are being achieved by various concentration of zinc oxide nanoparticles. This is the literature, what I am just summarizing that. So there are various bactericidal effects that is similar to antibiotics. Of course, the zinc oxide nanoparticles are very excellent, which have the ability to electrostatic interaction between the nanoparticles and the cell surface of the pathogen. That is most important. Once the cell surface has been intact or bind with the nanoparticles, there will be not much effect on the cells for its motility. What happens, during a certain period of time, the primed nanoparticles bind with the surface of the cells of the pathogen or the spores or the colony. It results in the enhanced cell damage. So this is most important when we can achieve easily the suppression of the pathogen, and at the same time during this process of suppression, the plant will be free of colonizing with the pathogenic system. So thereby the whole entire immune system of the pathogen will trigger or will have a betterment for its survival, as well as it will have its natural metabolic activity. Thereby, we will have a good amount of yield. So, as per the literature, zinc oxide nanoparticles also exhibit various antibacterial action with viability on biomedical surfaces, and also eco-friendly characters like rigidity, load, toxicity, stability, biocompatibility, and biodegradability. Most important, what now we are concentrating on the SDGs goals. Recently we had this G20 in India. It is a proud moment that SDG 6th goal says water and sanitization. So with these, whatever the elements which we are externally coating with the biological system in the environment should be a strong biodegradable. So that is the concept of using any technology. It should not harm the environment for a longer period of time. So biodegradable materials has to be introduced into the system. And in this way, zinc oxide nanoparticles, I'm talking about at very low concentration, it is naturally biodegradable, which can replace the natural fertilizers and can be used for enhancing the soil health. And this has been proved by the FDA, that is US Food and Drug Administration, that recognize 4 separate zinc compounds containing zinc oxide that usually classified as a GRAS, that is generally recognized as a safe material. So this is the concept where we use for more utilization of the zinc oxide material in order to synthesize the various nanoparticles by using various capping agent that we use for the plants. Yeah, of course, I am just targeting, or since I come from the plant pathology background, I am more emphasizing on the crop improvement as well as antimicrobial properties. But if you see the literature, zinc oxide nanoparticles, it's a versatile applications. See the application part. You can just imagine where else, what else it has left out. Almost all, in all the fields, zinc oxide nanoparticles can be used in the biosensors. It's very strange. You know, the biosensors nowadays used for the, detecting materials like quantum dots. These are all the materials they are used, and zinc oxide nanoparticles, they usually consider as a most important element. It can be used in the biomedical, cancer treatment, anti-diabetic activity, and also, as I said, it is mainly used in boosting the ROS scavenging enzymes as and when required at the spatiotemporal activity that is been under the stress when plants are exposed to various stress, and it is also used in the gene delivery. My target of the studies is it is used in the plant protection. So that is what I use. It's a sustainable practice in order to overcome the biotic organisms which are attacking the plant at various stages. Yeah. So when I talk about organism and the plant, what comes next is the tripartite interaction of the external elements that we are introducing into the host plant. In the host plant, there are various associated materials that will be there in the form of organisms, like rhizosphere organism or microbiome that is present near the rhizosphere, which may act as a carrier or a barrier in order to see that the plants remain durable, or can overcome, or it can suppress the pathogen, or it can mobilize the nutrients from the plant system, or it can secrete some beneficial molecules that is required for the plant during the various growth stages. All these elements, we call it a translocation and the biotransformation process that can be seen in the zinc oxide nanoparticles that is primed in the host plant, which acts as a plant innate system. That means once the material has been induced into the host plant, it will be remaining in the soil forever, and whenever the soil is under stress, which it requires, these particular zinc oxide nanoparticles will trigger the plant in innate host system so that it can protect themselves. It is a induced systemic resistance aspects where the plant will get more boost towards pathogen attack. So this is what the concept says. It can enter through the stomata, it can enter through the cuticle, or it can be used in the form of foliar exposure or through any wound or epidermal layer of leaf. So these are the major translocation systems that is been observed in the zinc oxide nanoparticles in plant innate system. And this particular mechanism or the system is been well proved in most of the cash crops. I mean, solanaceous plants, various work has been done, and this still holds good that it can protect the plant for a longer period of time until the harvest. This is what a particular a sustainable goal meets. So coming to the main topic, like the research which I am just emphasizing a small case study which has been done in our laboratory. We have used this Calicarpa tomentosa. It's a very good plant-based materials, and nowadays, more emphasis has been given to produce more plants, tissue culture plantlets, because these are conserved as a precious materials. And in some parts of the area, this plant has been becoming extinct because of its overuse, or the climatic change is also affecting this growth plant. So several researchers have reported the production of zinc oxide nanoparticles using various plant materials, starting from our gingiber up to your eucalyptus and the cassia. These are most commonly available plants that is been widely used, and they have found a fair amount to a promising amount of results when they check the plant materials either for the biotic stress or the abiotic stress and the biomass of the plant. It has given a good promising result. However, when we compare the genus Calicarpa has not been explored for its efficacy to synthesis in organic metal oxide nanoparticles, particularly zinc oxide. So this keeping the mind that, okay, all the other plants have been used and zinc oxide has given a very excellent results. Why can't this Calicarpa can be used for our studies? We have taken up this, or we have selected this plant. I don't want to stress more about on the methodology part, because I am sure that most of you are well versed with the nanoparticles, and I'm not so much expert in the instrumental or analytical part. Maybe some chemistry or physics professors are there in the resource as a participants. They may be knowing more better than me. Just for summarizing, we use these analytical instruments for characterization studies, and also see that these nanoparticles have a major element, what the component which we are using, that agent is present or not, and also confirming the size. That is most important. We have used various analytical materials for the synthesized zinc oxide nanoparticles to confirm it is nanoparticles. And these are the significance of the analytical instruments, where UV spec is used to see the formation of any nanoparticles. I have just given the selenium here. And DLS is used to measure the Z average size and zeta potential, where negative zeta potential ability of the nanoparticles will give high stability. This is what the concept says. And FTIR, of course, to know the functional groups associated with the nanoparticles. And XRD, powder XRD verifies the crystalline nature of the nanoparticles, because each zinc, silver, copper, and other kinds of they have a different nature of formation of nature is different. So study the surface, we use this STEM EDS to know the surface, as well as to some extent EDS will give the proper element that is been precursored in the synthesized materials. And of course, the HR STEM is used to definitely confirm the proper shape and size of the nanoparticles. This is the results of the UV in case of zinc oxide nanoparticles. Those who are a beginner, they can have a peak which is been expressed at 360, between 360 to 363 nanometer is majorly considered as the zinc oxide. So FTIR is the next is the FTIR we use to know the functional properties, and scanning electron microscope, where it says it has a range of 30 to 60 nanometer size has been synthesized that has been easily detected by the cell. Of course, we have also done the elementary composition of the zinc oxide. Here, the major peak that is been zinc has been produced. Other than that, we have also C and O is been implemented. That shows the synthesized particles are purely the synthesis of zinc oxide nanoparticles. Powder XRD, the diffraction index of the theta values majorly confirms that the significance of the nanoparticles will have more stability in the applications, and this is somehow we can say the particles are biodegradable in nature. And the TEM is been done to know the size of these nanoparticles, which is 31.76 to 82. That is the range we have got. And since the lower amount of size is considered as the best nanomaterials, it is the synthesized nanoparticles are more promising, and we have got a specific significant characterization data in our studies for taking up further for our other applications part. When I was talking about materials that has been more pathogenic in nature, and these are pathogens are highly virulent in nature. Wherever the crop, the host plant which is being affected in any of the stage, the Ralstonia is known to have it is a soil-borne pathogen, it's a seed-borne pathogen, and it is also known as a secondary infection. So that means in all the stages of the growth of the crop, this Ralstonia have the ability to infect the plant in various modes. So in order to know that, we have selected the most specific organisms, that is bacteria, Ralstonia solanacearum, which is most common in almost all the Cucurbitaceae as well as solanaceous group of plants. And also we have taken the Pseudomonas syringae. I think this is one of the model pathogen we use in order to check the overall control or overall ability of the plant by using the Arabidopsis thaliana plant, and this is the model pathogen they use for testing. So we have selected that particular Pseudomonas syringae pathogen, and also Xanthomonas campestris have been used. And if you see the elements of zinc oxide nanoparticles, as I was talking about, there is a issue, there is a debate in the future that nano toxins will be a major threat to the environment, and we will have major issues through these nanotoxins. That's why we are using at a very minimum amount, that is 100 ppm. When we consider the standard, that is also lesser, that is also more than the concentration what we set for the nanoparticles here in our study. And we use for Ralstonia solanacearum, I am talking about zinc oxide nanoparticles at 100 ppm. It has given a very significant inhibitions you can see here. And this is the standard where we have kept in the middle, and 37.2%, the least one we have recorded in the test sample. Likewise, for Xanthomonas, we have used 75 ppm, and for Pseudomonas, we have used 100 ppm, and for Campestris, we have used 50 ppm. So that means we are not crossing the limit. So there is a literature says nano fertilizers at the rate of 500 ppm is more safe as per the grass. But we are limiting our concentration, that is 5 times lesser than the recommended dose. So up to 100%. In some cases, Xanthomonas campestris, of course, it's a major problem in our paddy and also in cotton. But at 50 ppm, this has a very good amount of suppression. I mean, the most we have taken the moderately susceptible crops, and this is the amount of inhibition we have used. Other studies we have conducted that the in vitro antifungal activity of this pyracularia is a growing, emerging pathogen. If somebody is from North Karnataka here, I think you will be knowing more importance of this pathogen. This is causing blast disease in most of the cereals, including your, the millets, the rice, all other cereal crops have been majorly affected, and we have also selected this pathogen to see that what best inhibition can be achieved by the zinc oxide nanoparticles. And all these experiments we have confirmed in vitro conditions excellent. And why can't, once the problem is being achieved at the pathogenic side? So there is a issue that since nanoparticles cannot be used directly in the field level, okay, we detach it, we do some leaf slit assay by collecting the plant leaf from the host plant, and we incubate it, we treat the nanoparticles at different concentration, and we inoculate the pathogen on the leaf where we see various suppression level. And in our case, sorry, 100 ppm, 50 ppm are very excellent. So these, like similar to our, the concentration we are setting for the antibiotics, like we are setting for the minimum MIC, we are setting for the concentration for our nanomaterials, which is not going beyond the 100% or 100 ppm concentration. And 100 ppm concentration, when compared to the control, which is untreated with the nanoparticles, you see about 80% of the suppression can be achieved by the 100 ppm, and 50 to 60% of the suppression can be achieved by the 50 ppm concentration. So it is very excellent. And again, as I was talking about versatile application of zinc oxide nanoparticles, I mean a best biopesticides. That means we tested for pyracularia, we tested for Ralstonia, we tested for Pseudomonas, we tested for Xanthomonas. Now we also test the pathogen, that is Colotrichum, on the chili plant as well as tomato plant. We use the alternaria solans. These are most common pathogens, disease that is been a major threat for the farmers in the field level, and most important pathogens we have tested, and we have achieved a fair amount of good results in our study. And this is the first study in our case where we have observed zinc oxide nanoparticles at 100 ppm. This is a biotic factors where we are talking about Pseudomonas, that is Pseudomonas infestans. Zoospores have been completely aggregated, wherein the sporangia is not allowing any zoospores to release. These are all, in vitro conditions under the compound microscope we have observed, and to see the motility, how best it is been binding to the spores of the zoospores or the sporangia. We have also seen in the AFM, that is atomic spores microscopy, and also the scanning electron microscopy to see the binding capacity of the spores, where it can act as a electrostatic binding on the surface of the cells, where in that the zoospores or the sporangia that is present in the spores but is present inside the sporangia will not be coming out, so that the cells will be arrested. This is what it is very clear from our study. The fluorescent microscope images also we have tested for the major Ralstonia to see that live and motile and dead cells of the bacteria can be seen. And in the, you can see the green spots here represent the live bacterial cells, and the red spots here indicate the dead bacteria. So that means the capability of the survival of these pathogenic organisms after treatment with the zinc oxide nanoparticles in the in vitro conditions, while tagging with some fluorescence, is very clear that the motility of the bacteria will be lowering and coming to the death stage. This is what we observed from our study. The cell wall changes, and it also observes the shrinkage of the non-homologous surface, and it finally will collapse and it will die. So coming to the in vitro conditions, antimicrobial, excellent. Then in the leaf assay, we implant colonization we observed. Yes, it is very effective. Then we also test that measure how mechanism it is working, whether it is interacting, binding the cell wall surface. That is also been observed by various experiments we have done. Now the time is to application. Whether antimicrobial in vitro is successful, whether this zinc oxide nanoparticles can be used for treatment, either is as a seed priming or can be used as a spray. So this is what the experiment we designed. We take the moderately susceptible seeds, we treat the seeds with the zinc oxide nanoparticles at low concentration, that is 100 ppm concentration, what we have observed for the in vitro activity. Then we raise the plants, treated plants after a certain period of time of 21 or 3 weeks of time, we inoculate the pathogen. This is the late blight pathogen we inoculate, and we check the early emergence, whether in comparison to the control, what best it can give, whether it can produce early emergence, increase the plant height, and also early flowering. This is what now we are looking for. A shorter period of time, 90 days of crop, whether it can be reduced to 75 days, whether it can be reduced to 80 days, and if it is reduced, whether the yield can be improved, whether it can be, the biomass of the plant can be enhanced. This is the concept of sustainable crop improvement, and we are trying to work on these aspects, and by using the zinc oxide nanoparticles, we have got some fair amount of good results, and further we are planning to implement in the large scale. This is how we treat with the seeds are treated with the zinc oxide nanoparticles. This is the treated, and that is the untreated one. This is the results which we got for our early emergence. Of course, the seedling vigor, plant height, number of days, in compared to the control, it's about 9 days, 7 to 9 days, we have reduced the flowering period. That means not only the flowering period, comparatively the yield has been more enhanced in the treated one compared to the control. So lesser the time, more the yield. That's what we need in the sustainable development program, and that is what we are working on it. I was talking about systemic nature of the zinc oxide. So how to prove that? So we have inoculated the pathogen at spatiotemporal level. That means grow the seedlings, inoculate the pathogen at different time intervals, I mean different days intervals. So 1 day, 3 days, 5 days, 7 days, 9 days. So it's a gap of like 10 days, we inoculate the pathogen. That means pathogen is continuously under the stress. We are inoculating the pathogen. And those plants have been raised by the treated one. And if we see the symptoms appear on the plant surface, we again go for a spurious, the spray application, where the zinc oxide nanoparticles can able to suppress the late blight. This is what our observation, and through our experiment, you can see the primed plants with the pathogen has shown a very good amount of biomass as well as a greenery compared to the pathogen as well as the control plant, and this have a major effect on the yield. And this is what we have observed in the spatiotemporal induced systemic resistance in the greenhouse conditions against late blight of tomato. This is the mechanism I told you once it is been proved. Sometimes what is the mechanism is the biggest question. Someone is asking about, okay, it works. You know the chemical. Chemical knows how it will work, how it will bind. We know that. What about your technology? You are talking about it is a feasible, it is a eco-friendly, everything. How you are telling eco-friendly? Whether it may have some adverse effect in the future. No, it will not have any adverse effect. That's what we say. It induces into the host in the innate system. That means no other entities are been allowed here to promote the plant growth to suppress the pathogen. Here the plant itself will boost its induce or it will boost its metabolic activity, physiological activity in terms of cellular level, in terms of biochemical level, and in terms of this transcriptome level. That's why we have used the cellular defense biochemical transcriptome level, where we use callose deposition. We have checked lignin, hydrogen peroxide, and also the enzymes we have tested. These are all scavenging enzymes that is mostly present in almost all the plants when plants undergoes the stress. So this is what we observed: increased enzyme activity, and also at the transcriptome level, the most significant unigenes which have co-expression factors. That means plant induce the resistance, plant promote the growth. And plant promote the growth in what way? Sometimes it says upregulation of ABA, sometimes upregulation of the pigments. This is what the plant does inside the plant system, and it is not been observed outside. That mechanism has been proved by using the plant which have been treated with the zinc oxide nanoparticles. This is the methods which we use for our histochemical deposition to see at the cellular level. At the biochemical, we use these three major core enzymes for checking. And this is the results of the histochemical. See the deposition here in the treated as well as in the pathogen inoculated well. This is the control one, no deposition. That indicates very clear. Even in the hypersensitive reactions also can be observed at the seedling level, which also can be used as a marker that can be screened for screening the resistance as well as the susceptible. That can be used in the breeding program. This is what our intention is. So research we do in the lab, okay, it is done. This can be used as a marker. If it is used as a marker, definitely it is a great contribution to the society, where the breeders can use our markers to screen the various germplasm and can deploy or can breed resistant varieties through our markers. This is what we are attempting in the present situation. This is the callose deposition. Various time intervals. See at 24 hours, it's very boosting the signaling is very boosting here. Same as with the enzymes, log 20 initiated at 12 hours, upregulated at 48 hours, and slightly decreased at 96 hours. So this is the concept we use for our glucanase, SOD, and other things. And also, as I was talking about, this we have selected from differential expressed proteins in primed and non-primed proteins. Also we studied, where we have done the 2D gel electrophoresis to identify the differential expression of the proteins that are majorly triggering the resistance as well as the promotion of the plant. These are the genes or the primers which we used our study. This is the hypothesis where we use it very clear that the zinc oxide nanoparticles plus the pathogen at these time intervals has a very excellent result. And coming to the last point of the study, utilization of plant resources, how best we are utilizing and in what way you are utilizing, whether it is a feasible, affordable, whether it is a eco-friendly approach. Through some of my slides, I am quite sure that the results which we have, the experimental setup which we have done, to some extent, most of the synthesized nanoparticles which can be used at the minimum concentrations are very excellent. Of course, not only with multiple properties to exhibit in the plant system. What we observed is broad spectrum activity also we observed, that is the zinc oxide nanoparticles synthesized by using the Calicarpa tomentosa, and which is very much correlating with the literature that is available in the other plants which they have already been done. And for the first time, we have demonstrated the aggregation of the Pseudomonas, that is biotic pathogen, very tough to handle, tough to maintain, tough to isolate. And these are very vigorous, very virulent pathogens. Pseudomonas infections is the one which created a history, that is late blight of potato, the Irish famine story. Nowadays, this same strain pathogen is also causing major issues on tomato and other crops. And we have observed that the aggregation of the Phytophthora infections, particularly those spores, are when we treat with zinc oxide at nanoparticles with 100 ppm. And zinc oxide nanoparticles in the implanta colonization, as we observed, at any point of time, whenever there is a secondary contamination on the leaf surface, plant definitely will undergo stress, will produce the symptoms, and it is difficult for the farmer. If he knows in the early stage, he can uproot it, he can just destroy, he can burn it. When the plants are at the verge of giving the yield, if pathogen attack, if the symptoms appear, so it's a problem. So for that, to prove that these zinc oxide nanoparticles can act as a curative, can act as a protective, can act as a systemic in nature. All three sets of experiments we have demonstrated, and from the curative activity, the leaf assay we have done clearly demonstrate, treat with the nanoparticles, spray with the nanoparticles, wherever the secondary contamination plants will be explored or exposed to attack, definitely it will have certain amount of protection. This is what we observed, and the priming with the nanoparticles with the seed material have a very positive effect on the growth and development of the post-tested plant we done with the tomato. And zinc oxide nanoparticles induced plants particularly resistance up to 72.9. This is very excellent where we observed for the late blight of tomato disease. And this induced resistance is also very well correlated with the corresponding alterations at all the three levels. That is the innate system. Pathogen attack at the cellular level, yes, tested. We know it will suppress by triggering some cellular defense mechanism. We call it as first line of defense mechanism. We observed the lignin, very good deposition. Callose, we observed H2O2. These are the major biomarkers at the cellular level. It will trigger in the host plant, suppress the pathogen. Biochemical, particularly the scavenging enzymes we have tested. PAL we have tested, peroxidase, LOX. These are very excellent bioenzymes. Though the pathogen is clever enough or vigorous enough to enter into the cellular system, but the next line of defense in the form of biochemical enzymes will not allow the pathogen to colonize inside the host. So it will have its own defense mechanism will be attributed, and it will suppress the disease. And finally, if the pathogen comes across histochemical, overtake the biochemical, it leads to the genomic level. That is the genetic material. The gene of differential genes will be activated, transcriptome level, and the defense genes will not allow the pathogen to colonize into the host plant. And at the point of time, it will make sure that the pathogen or the plant will undergo the HR expression. And also, all these cycles have been completed, and we are more curious to know what are the proteins that is been involved, which is more functional in nature for triggering the resistance as well as the triggering the enhancement of the yield. We have, from our results, we have got 5 differentially expressed proteins detected that play a vital role in the establishment of plant defense. So just a word of conclusion. Green zinc oxide nanoparticles, I personally consider it as a biomolecule with futuristic applications for triggering crop immunity. If you go to the ICAR website nowadays, more talk is on zinc oxide nanoparticles, because as I was mentioning you, there is a guideline from the GRAS that is replacement of this with nano fertilizers. Nano fertilizers with multifaceted properties. If it is a co-expression factors like nanopesticides with nano fertilizers, definitely the crop can be protected in all this manner against various foreign organisms or any stress, including the environmental stress that is being exposed to the plant, so that the plant will not be affected when the plants were either treated in the form of a priming, either treated in the form of a soil treatment with the zinc oxide nanoparticles. Definitely, we can protect the plant. We can protect the genetic resource of the plant material that is available, and I am sure that this is a very excellent material for the future application for triggering the crop yield. So I think this is what I was planning to talk about in today's talk. So I'm very much thankful for the organizers, and also my personal thanks to all the participants of this refresher course who are being here to witness my talk. And I once again thank all of you for your patience hearing.
Sir, I have
Thank
one
you.
question,
Thank you
sir.
very much. Yeah,
Sudhi-
any questions,
Sudhi-
you are most welcome.
Yes, sir. Yes, sir. Sudhish, sir, I am Somlingappa from Gadag, sir. I have one question, sir.
Yeah.
Sir, are you hearing my?
Yes, yes, yes.
Sir, actually, today in the market, urea, that is nano urea
Yes.
available for the farmers, can be used as a fertilizer to improve the crop in their field, sir. Actually,
Yeah.
my question is that then zinc oxide, zinc oxide nanoparticle based the fertilizer, when it will be available to our farmers to get a higher yield and then to improve their crops, sir. Maybe in the future, I think actually,
Definitely. That is what definitely the ICR has formed a separate community under the NATP programs, where now, you know, as I told, I am not the new one to do this research. I'm just a beginner. These zinc oxide nanoparticles have been practiced from a long time. I have the
Yes, sir.
literature from 1996.
Definitely, sir. Yes.
1996. So, as I was explaining you, the issue with the bureaucrats as well as issue with the environmental nanotoxins issue, this was not been allowed for the field level. However, as you mentioned about nano urea and also nano composites also available. I think you may be knowing that.
Yes, sir. Yes, sir.
Nano composites also available. So the thing is, zinc oxide nanoparticles with nano urea or nano composite material naturally been synthesized. These are under the practice in the laboratory conditions, and very good results have been obtained.
Yes, sir.
The thing is, now it is under the coordinating trials, all India coordinating trials. You know that in
Yes, sir. Yes,
different
sir.
agriculture stations, we have various centers where they are exclusively working on these materials. And because it's not a 1 or 2 year results we can give, because mostly when I apply to the field conditions, I should look for the soil health. Whether it
Yes,
is
sir.
all these factors are a major task for the scientists, and they are deeply studying. And if it is at the lower concentration, I am sure it will be in the nearby years. I think another 2-3 years, definitely it will be in practice. You know,
Yes,
I was in Brazil, exclusively zinc oxide nanoparticles. There is a person, if you can just search in the literature. Professor Leonardo Frutiso is the topmost scientist, and he is the person who has contributed the major role in this zinc oxide nanoparticles. And in their country, they are using replaced with all the chemical fertilizers.
Yes, sir. That's why I am asking, sir. I am also, I have got very curious about this zinc oxide based nanoparticle based the fertilizer,
Yes,
sir.
yes.
It will be a very great help for our farmers to get a higher yield in their crops, sir. In their field, sir.
That is what we expect. We are also expecting, and our results are very clear. It is there, but
Yes,
and also see, as very honestly, if I go to the field, see, these are all we done in the controlled condition. Experiment than in vitro. Means laboratory conditions, everything is controlled.
Yes, sir.
The greenhouse condition, everything is controlled. But
Yes,
in the nature, in the field, it is out of our control.
Yes, sir. Yes, sir.
Only we believe our product, but its performance really depends on the environment. But I'm sure I showed 79%, 72%. I am sure in the natural condition, in the field conditions, definitely it will be 50%.
Hmm.
50% without any chemical. And you know, these are all feasible. There is very less money for this.
Yes, sir. Sir.
Very,
Before
very less.
before release them to the field, sir. Before to release them and make them to available to our farmers, they must be tested for eco-friendly and then sustainable.
Yes.
Other eco-friendly. They must be eco-friendly, sir. They must not harm to other crops, other fields, other
Yes.
plants as well, sir.
Yeah, that's what our aim is also.
Huh.
That is why, you know, we tested for versatile nature, because some are, you know, now we are going for traditional method of practice where we go for alternative cropping, mixed cropping.
Yes, sir.
So those practice maybe the progressive farmers who are looking for, you know, money nowadays difficult because too much of expense, too much of, you know, inputs are required. Instead of that, go for these kind of products which can take care of its own. Any kind of crop, apart from the unwanted crops, you know, we should take care of this application. With the recommended dose, since we fix ourselves, the packet will be available only with 100 ppm concentration. They cannot do anything more. Only thing is the mode of application, how many times they are using, that should be in a proper manner. I was experimental in my study in the greenhouse conditions. I did some priming the material and I allowed the plant to grow. For some extent, plant was protected. Afterwards, again the plant got infection. Then I thought, okay, the infection came because of
Yes, sir.
secondary contamination. That is through other source. What I did, then again we go for one more spray. So this kind of, you know, technical knowledge if you provide to the farmers, definitely they can apply and they will have a very, you know, good results, very good yield they can obtain from these plants by treating with zinc oxide nanoparticles.
Yes, sir. Sure, sir. Thank you, sir. Thank you, sir.
Hello, sir.
Sir,
Hi, sir,
good
please,
afternoon, sir.
Hello, sir. Can you hear me,
Yeah,
sir?
yeah. Yes,
Sir,
yes.
Venkatesh, Assistant Professor, Downgrade University, sir, for Technology Department.
Yeah.
Nano zinc oxide nanoparticles induced plant resistance against late blight diseases. That is only for only one generation. Also, in greenhouse condition only, show a in first generation it show a protected against the diseases, resistance against the disease. The next generation
Yeah, that
in
is
green.
what. No, that is you know, that is why we have done the spatio-temporal analysis here. I was telling you, pathogen only attacks one time. You know that. It's a biotic. It's a living organism. Once it infects, it will start colonizing the host plant. For that reason, what we did, we made a setup of 10 setup of plants where we inoculate the pathogen. Artificial inoculation was done at a certain period intervals. So in this way, what we did, we saw the durable resistance, checked with the symptoms that is appearing on the host plant, as well as mechanism also that I have explained. Here, the point is the plants which produce the fruit that we need to harvest.
Yes.
Harvest materials again should be recycled, again should be applied, and it should be grown separately and inoculate the pathogen and see its effect.
Okay.
That we have not done. We have not done. At the same time, since we tested its durability at the genetic level, we have done.
Yes.
Genetic level we have done. That means, see, any kind